Good afternoon, ladies and gentlemen, and I'd like to thank Chris and Martin for the kind invitation to come and talk to you about a subject dear to our own hearts in UCD on the research front. So for the MSc students in the class, now you have to pay particular attention because this talk could end up on an exam paper. Okay, so I wanted to talk to you about the emergence of resistance to antimicrobial compounds, and then I thought I'd put in these three little riders: opportunity, power, and usage. Opportunity to mean that bacteria may share different environments and therefore the potential for transfer is increased. Power meaning that they have the ability or the enzymatic wherewithal to actually stitch in new genes, and usage meaning that they're able to decode that new information coming from foreign genetic backgrounds in order to make use of the new genes that they acquire. All key parts to the story of resistance. So in the talk there are 4 pieces to it, which I will go through very quickly. The first just again describes some of the background, but again Philippe and others this morning have very nicely covered that, and I was particularly impressed by Katie's talk. For somebody so young, she certainly had a very good understanding of what resistance was. Then I'll take you through some examples of the molecular characterizations, and I'll show you this interaction between animals and humans using genetics to do it. And then I'll come back to this idea of opportunity, power, and usage, and try to show you how we may be able to predict the emergence of resistant bacteria. And then I'll try to end up with 2 examples close to public health and some conclusions. So without further ado, let's start off and talk a little bit about the use of antimicrobial compounds in veterinary medicine. Well, as Philippe defined this morning, these compounds are very, very valuable indeed, and they are naturally occurring in many cases, and they're designed by nature to kill or inhibit organisms. But again, as Philippe pointed out, the sort of plethora of drugs that we currently have are really all that are, they're all that we have in existence at the moment. And essentially, we've no new drugs discovered in the last 30 years or so. So therefore, protecting the efficacy of the existing arsenal is critically important. Now, Bjorn in his talk this morning also mentioned this strain Streptomyces venezuelae, and this is an example of one of these antibiotic producers, a naturally occurring strain, which also has an inbuilt resistance mechanism. So we then have the resistome on the right-hand side of this diagram, which is the totality of either natural producers and then associated with those natural producers, the resistance mechanisms that are genetically geared and inbuilt into those systems, which we now need to begin to understand. And the last thing I'll say about this slide is that the same antibiotics are used in human medicine, animal medicine, and to treat plants. Now, WHO are really concerned about this, and they've come up with a sort of a guidance list to try to focus our attention on drugs that are critically important, drugs that are highly important, and then all other drugs. And then they've indicated a category of high priority drugs whose efficacy we really do need to preserve. So on the bottom part of this slide, you see that the 3rd and 4th generation cephalosporins are very important, as are the fluoroquinolone drugs, the macrolides, and the glycopeptides, which we've only heard one mention of so far today. So in terms of the resistance mechanisms that I'll talk about, I'll really focus on the 3rd and 4th generation cephalosporins and the fluoroquinolones by way of example. Again, Philippe covered this, but he showed you that this is a typical bacterial cell, and in the bacterial cell you have very unique targets, and that's why the antibiotics are so valuable, because we describe them and define them as being selectively toxic, as opposed to a biocide, which would be non-selectively toxic. So this selectivity is what gives them their very important pharmacological value. And when you get resistance at any one of these bacterial structures where there's a modification, usually that resistance is linked with a gene or a number of genes, which then can encode that resistance and spread that resistance mechanism to other bacteria. So what is resistance? Well, we must be able to measure it. And really we use kind of epidemiological cutoff values as a guide to start measuring what we talk about in terms of resistance of bacteria. Now, on the left-hand side of this panel, you'll see what are called wild-type bacteria. So these would be organisms which I could just dig up in the garden outside and just measure their minimal inhibitory concentration to a particular antibiotic. And what I'd expect to find is that I get a random distribution, a typical distribution of the MIC values. When you look at the epidemiological cutoff at about 0.125 milligrams per litre on the right side of that left panel, Then when you go above that value, you now start to see what are non-wild type strains. So these may be bacteria coming from clinical scenarios where their MIC values are very different to the epidemiological cutoff. And these we define as being either susceptible broadly, intermediately resistant, or completely resistant. And of course, if an organism is completely resistant, that means that that drug cannot be used to treat such an organism involved in an infection. So let's now try and imagine in our minds what happens when a bacterium is presented with the stress of an antimicrobial compound. Well, I use this very simple analogy of a bacterial culture. So here's a bacterial culture happily growing. So long as there's enough nutrient, the temperature is okay, the light is okay, this continues to grow. And on occasion, we see the emergence of a mutant. My pointer, I don't think is working very well, but you can just see the one I'm talking about right in the middle. And so long as there's no selective pressure on that mutant, the culture continues to grow and the mutant might disappear again and everything is hunky-dory. But now, when we take the same scenario again, impart a selective pressure, now the mutant appears, and when the selective pressure is in full force, then instead of the normal wild-type bacteria, they tend to disappear, and the mutant population starts to come to the fore, and it starts to outcompete the wild-type population. So this is, as I say, a very simple scenario. So if we remove the selective pressure again, we may see the removal of the mutant, although it's not guaranteed, and it really depends very much on the type of mechanism that's involved. Now, there is one other example of this. Well, in fact, there's two other examples, one of which I'll come to now, which is the collateral damage associated with the microbiome. So the microbiome is the totality of bacteria in an animal's gastrointestinal tract or in a human gastrointestinal tract, and the impact of using a drug on that microbiome. And this shows it to you very nicely. This is a study published about 2 years ago, which looked at what's called the virome. So this is the totality of all viruses in that particular ecological niche. And here you'll see on the top part of the slide a couple of examples of different viral families which were noted in this particular study. On the bottom part and the left of the panel, you see a phylogeny of all of the organisms or viruses that could be identified within that particular animal microbiome. And what I want to particularly draw your attention to is the right-hand side at the bottom of the panel, where you compare non-medicated versus medicated animals, and you look at the virome over time. And what you see from the right-hand side of that panel is an amazing shift in the virome as a consequence of the selective pressure being placed by the use of a drug. Now, we went back to the database when we saw this paper, and we started searching the database immediately for all known genomes of viruses that we could find. And so on the left-hand side of this slide, you see the different bacterial families. In the middle, you'll see the viruses that we could recognize from the genetic information. And on the right-hand side, rather unsurprisingly, you see that in many cases, there is an antibiotic resistance gene or an antibiotic resistance mechanism associated with nearly all of those viruses coming from different bacteria. So viruses are particularly important in terms of the transmission of resistance genes. That brings us then to the amplification of this process. And in my way of thinking, I think there are two approaches to this. The first one is a clonal expansion of resistant bacteria. This is where we amplify a particularly well-adapted strain, maybe the kinds of strains that Bjorn was talking about this morning in his talk. But there are three examples here. One is this pandemic Salmonella typhimurium DT104 strain, which is a penta-resistant organism. It's simultaneously resistant to 5 different antibiotic classes, and I'll come back to that in the end. Campylobacter was one that was mentioned, and then methicillin Staphylococcus aureus, which again is a major problem in the hospital environment. And all of these organisms are well adapted to their particular ecological niches so that an antibiotic selective pressure could see the expansion of these populations. The second possibility here is the involvement of resistance markers or resistance genes. And here we have to take note of things like conjugate of plasmids, which I'll come back to later, ICE elements, and ICE here doesn't mean in case of emergency, it means an integrative conjugative element. It's a very large repertoire of genes, and bacteriophages, which I've just mentioned. And inserted in all of those 3 structures, there are other elements such as insertion sequence elements, transposons, and integrons that play a role here. So what is the problem then? Nobody has said what the problem is. We've talked about veterinary medicine, but we need to try to boil this down. I think the challenge for antibiotic resistance is different if you talk to human medicine people as opposed to veterinary people. In the case of human medicine, the issue here is the emergence of superbugs, such as MRSA, methicillin-resistant Staphylococcus aureus, or Visa strains, not referencing your credit card here, but rather vancomycin, intermediate Staphylococcus aureus. And vancomycin is an example of a drug which really is the last line of defense. It's a glycopeptide, which is a very valuable therapeutic agent, which we are now seeing resistance emerging to. Then in human medicine, we also have the role of gram-negative bacteria, which I'll come back to. The fact that there's no new drugs in the pipeline, which is a major problem, as I've said at the outset. Hospitals and nursing homes are major reservoirs of these resistant strains. And then there's this zoonotic and commensal link. And John mentioned the commensal link, and nobody was picking up on it. But the commensal link, I think, is like a smoking gun, because I think here you could see quite a lot of resistance genes beginning to populate. And I'll show you an example of that in the end. And then you have foreign travel as a risk factor. On the veterinary side, it's not such a major problem. The situation has arisen because of the deteriorating situation in human medicine, and that's why the veterinary profession need to take action here. In terms of veterinary medicine, the issues for them are infections in humans caused by the transmission of zoonotic bacteria, or the transfers of these resistant bacteria or the resistance genes via food, direct contact, or through environmental means. And then the third element is, what about the environment? We know very little about the environment, yet these three domains are interconnected. So we see bacteria moving from animal domain to the human domain via the food chain or by direct contact, and similarly from humans to the environment or vice versa, animals to the environment or vice versa. So surveillance is becoming an important issue, and you'll hear more about that in the next talk. Now, what about the molecular characterization? Is it always the same genes? Are there different genes that are moving? Let's have a look at that. Again, Philippe showed you resistance mechanisms in his talk this morning. And this slide really distills down into the four major mechanisms that are associated with resistance. You have enzymatic drug inactivation, which is typical of betalactamase enzymes, and we heard from Katriana's talk this morning about the betalactamase issue. Drug modification or replacement, which is typical of fluoroquinolone resistance, and then active efflux or reduced drug uptake. All of these four mechanisms are governed by one or more genes that are corresponding to those mechanisms and giving rise to resistance to one or more classes of antimicrobial agent. And once those genes emerge, they can be transmitted either by direct contact via conjugation in the left-hand side of this, or by transduction, which I've already mentioned, or by a little understood mechanism called transformation, where you see the direct uptake of naked DNA by certain types of bacterial cells, one of which in this case from a zoonotic aspect is Campylobacter. Now, just to give you a little bit of research background here. This is a study which illustrates the importance of surveillance on commensal organisms, and I'm really glad to hear John mention this morning, because I think surveillance of commensal bacteria is critically important in order to guesstimate a value of how relevant they are. This is a study we did with the Department of Agriculture, Food and the Marine some years ago, where we took a broad collection of about 100 different multi-drug resistant bacteria, many of which we thought were commensals, and in fact most of them are, not all of them. We then assessed their antibiotic resistance profile by conventional methods and then genotyped them. So on the right-hand side of this slide, you see the phylogenetic information, and the ones to look at here are the A and the B, A and B1, and A and B1 signify that most of this collection is actually a commensal collection. So we worked on this in more detail. And basically, to cut a long story short, the upshot of this study was we saw a lot of resistance to the older drugs, so tetracycline, streptomycin, sulfonamides, and ampicillin. And again, that's not particularly surprising. Those drugs have been around for a long time. Bacteria have had a long time to adapt to them, so that's not particularly surprising. But what is surprising is that when you look at the panel on the right-hand side, you see two classes of drugs, the aminoglycosides and the betalactams, which are on the critical list from WHO, and we see quite a number of genes which are giving resistance to these two classes of agent. When you look at it in more detail, again, the top part of this slide on the left side, you see the strains that are at issue. You see the donor resistance profiles here indicated to you in shorthand notation. And if you take it from me that in most cases, those complete resistance markers can be transferred by conjugation to other bacteria that were once then susceptible, converting them into a multi-drug resistant strain. So this movement of genes via conjugation through bacterial populations is particularly important. And when you stress an organism, you can actually drive that effect much more efficiently. And the bottom part of this slide just shows you an example of an integron, which is a genetic element which is responsible for part of the makeup of these plasmids. Now again, coming back to these extended spectrum beta-lactams, you've heard some mention of this during the morning, but one of the key issues here is their role, and particularly in terms of transmission between bacteria of food-producing animal origin and then their potential role in public health. This is a study published just last year, which was an analysis of a large collection of Enterobacteriaceae looking at the prospect of extended spectrum beta-lactam resistance. And A, B, C, and D represent different food-producing animal populations with showing you different segments of the pies indicating the different types of extended spectrum beta-lactam gene that you can detect. E is the human. Now you see that there is some overlap between certain food-producing animals and the human, and it's very important now that we try to define this and try to understand what is going on so that we may be able to develop mechanisms to control it. This is a heat map which shows you part of that study. On the left-hand side are all the bacterial strains that were studied, some from human, some from chickens, some from cattle, some from pigs, and some from lambs. That's on the left-hand side of the panel. They all contained large conjugate of plasmids, and these conjugate of plasmids carried one or more resistance markers to these ESBL drugs. All these strains were multi-drug resistant, and they contained different populations of plasmids. We studied a number of these plasmids in detail. So these are the sequences. This is the total sequence now of two very large plasmids coming from the same human isolate. And here you see two different plasmids, one of about 166,000 base pairs, the second one of 107,000 base pairs, two different incompatibility types, and the red arrows in the plasmid structure indicate the resistance genes. And you see rather remarkably in these structures, there's very little else in the structure, quite apart from the resistance elements themselves. So that's a rather bizarre development which we don't quite understand just yet. Same situation in this chicken isolate, again, two large plasmids which came out of that. And in the case of these two plasmids, on the one on the left, you see a gene called CAT-A1, which encodes fluoroquinolones resistance. And on the one on the right, down at about 4 o'clock, you see a gene called BLAST-CTXN1, which is an extended spectrum beta-lactamase encoding gene. Again, to cut a long story short here, this is just a summary of 16 of those plasmids which we've just completed the sequencing of to try to identify their structures, see what genes they have. And as I said, for the most part, they contain these ESBL markers in addition to other resistance markers. But what's rather interesting is if you look at chicken 49 and chicken 60 in the table, those are two different bacterial isolates, yet they contain the same 108 kilobase plasmid. So it just shows you that these plasmids are moving between different populations of bacteria in different ecological settings. More recently then, and again in collaboration with our colleagues in the department, we were able to identify as part of a study a very unusual type of extended spectrum beta-lactam encoding gene called a BLAST-CMY2 gene. Now this one is very, very interesting because this is kind of, again, one of those last line defense agents. And in this case, these genes, these CMY2 genes, which are indicated in yellow here, are normally present as part of a large genetic element that moves as a composite structure. But if you look at type 4 down below, type 4 comes from an Irish horse. And in this case, we see the same gene in a completely novel genetic context, which has not yet been reported. And this is, again, very worrying, which it suggests to us that there's an alternative mechanism by which this gene is moving around. Fluoroquinolone drugs were also mentioned, and in the case of fluoroquinolone drugs, there are several different mechanisms of resistance here. The major one is the one which Philippe mentioned this morning, which was related to this enzyme DNA gyrase. There are a couple of genes which encode that enzyme called GyrA and GyrB, but apart from those, there are other potential mechanisms whereby you can give rise to fluoroquinolone resistance. Fluoroquinolone resistance is sometimes related to foreign travel. And this is a study which looked at salmonella and foreign travel, where these individuals were coming back from very nice parts of the world, foreign exotic locations, and they were presented with gastrointestinal illness effects. And from their stool, Salmonella Kentucky or Salmonella virtua were cultured. When those strains were analysed in detail, the resistance pattern showed a commonality, which was a reduced susceptibility to a drug called nalidixic acid, the progenitor quinolone from which the fluoroquinolones are derived. If you look at the MIC values in the middle of the table, to nalidixic acid it's greater than 256 micrograms per mil. And in the case of ciprofloxacin CIP, you see a reduced susceptibility phenotype here. When you investigate this in more detail, in one of these examples, the one which I've boxed with the red broken box, We identified a plasmid which was very unusual, which showed us the presence of two interesting genes. It's shown to you here on the bottom panel in the middle. The BLA-TEM gene at about 10 o'clock encodes resistance to penicillin, whereas the QNRS1 gene encodes the reduced susceptibility to fluoroquinolones. So here you have a kind of a coalescence of two different classes of antibiotic resistance encoding gene, one to penicillins and one to fluoroquinolones. We looked at this in more detail, and in fact, when we searched the genetic databases, if you look at the PVQS1 line and you compare that with the PINF5, PVQS1 comes from a Salmonella virchow strain. The plasmid from PINF5 comes from a Salmonella infantis strain. The commonality between those two isolates is that they came from Thailand. So here we have a situation in Thailand where probably originating from an avian or some kind of a bird, this plasmid emerged, moved between different Salmonella strains, and using this kind of forensic genetics, we can actually pick up the relationship. The last example I'll give you is of what's called an integrative conjugative element. Now, the people dealing with MRSA will probably know this because MRSA, the gene for MRSA, which Philippe alluded to, called PBP2A, moves as part of one of these ICE elements. We see another example of these ICE elements in Salmonella DT104, which I mentioned earlier on. And in this case, in Salmonella, all of these genes that encode the penta-resistant phenotype are clustered in the same location on the chromosome. And it's called SGI1, and that SGI1 island moves en bloc among other serotypes of Salmonella, such that you get this amazing array of resistance building up within this genus of the bacteria. Now that's all rather worrying. So we need now to try to figure out if we can begin to predict this. So can we recognize the emergence of these organisms using this idea of opportunity, power, and usage? Well, one of the certain developments in the last 5 years or so has been the capacity to do whole genome sequencing. And when you do whole genome sequencing, you really get a very detailed look at the genetic blueprint of a bacteria. So much so that we can begin to understand the emergence of resistance when we start to look at what we call sympatric bacteria versus allopatric bacteria. And sympatric bacteria are typical of the kinds of organisms we're talking about, Salmonella, E. coli, and so on. And these have a propensity to exchange genetic information when they're within a particular reservoir. They exchange genetic information by this horizontal gene transfer mechanism, which is this conjugation mechanism that I referred to earlier on. And this really is an example of true genetic pluralism. So for example, in this Enterobacter aerogenes plasmid here, here's a plasmid with all of the genes demarked in different colors, and all of those genes then map back against different genera of bacteria. So it's giving you a kind of a genealogy of where those genes come from and how they have come to be present in one plasmidic structure. We wanted to test this hypothesis. So we took a random collection of zoonotic bacteria, so E. coli and Salmonella, and more traditional nosocomial bacteria, Acinetobacter baumannii, Klebsiella pneumonia, and Staphylococcus aureus. And we specifically looked at the plasmids in these structures here, and we were looking for the presence of codon usage, so the codon usage patterns, which decodes the genetic information to give you a protein. So we built this in terms of a heat map where we measured the polarity of the codons going from left to right and the different plasmids here. Now, I think you'll agree there's probably not terribly much that you can see with respect to this heat map. So if you redraw the heat map using principal components analysis, you can see that we do get a division in terms of these bacterial genera. So much so that the zoonotic agents appear to map here, the nosocomial agents appear to map here, and the zoonotic agents appear to map here, with the exception of Klebsiella. That's an interesting one, because Klebsiella wouldn't necessarily be regarded as a zoonotic agent, although we do know that it can cause or can be involved in cases of mastitis. It's one of those organisms which is kind of like a smoking gun in a sense, because it is certainly multi-drug resistant, it has had clinical implications, and if it has these abilities of opportunity, power and usage, then it's one we definitely need to watch. If we redraw that diagram and we look at it in terms of nosocomial and zoonotic, you see that there is a clear demarcation between the ability of these organisms, whether they're nosocomial or zoonotic, to impart this power, opportunity, power and usage phenomena. So where does that all leave us? Okay, what's the impact then on public health? Well, again, you've heard the comment on the use of residues. And I think MRLs and measuring residues is particularly important from a food safety perspective. This is a very nice study which was done some years ago by our colleagues at UCC along with the Danish Technical University, where they looked at different antibiotics, so oxytetracycline here, erythromycin and penicillin, all of which are licensed for veterinary use, and they looked at fermentation mixtures. So these are mixtures of bacteria that are used in various types of food fermentations. And they measured, if you like, the impact of increasing drug concentration as you go across on the bacterial population. And I think, as you can predict, as you increase the drug concentration, the numbers of bacteria die off. That's a particular typical response. What you also see here is on the top parts of these graphs, the pH starts to rise, and that's very telling. In other words, when you have residues or active antibiotics in a food fermentation where there are live cultures, instead of the fermentation happening in the manner which we expect it to happen, the antibiotics reduce the concentration or the numbers of the fermentative bacteria such that they open up the opportunity to pathogens to flourish within this environment. So taking that information on the left panel and just reassessing it here using a fluorescent marker. So in this part of the experiment, what they did was they sunk in a fluorescent marker into the chromosome of E. coli 0157 and repeated the experiment. So in the absence of any antibiotic, the fermentation went normally. Sorry, it's the bottom one. The fermentation went normally. In the presence of the antibiotic, the fermentative population was wiped out, the pH rose, and you see the emergence of the pathogen by way of the fluorescent signal here. And this is, we think, what can potentially give rise to foodborne outbreaks associated with fermentative food. Not alone that, but there is a major impact which we quite yet don't understand in terms of the human enterocyte, going back again to the microbiome in the gut. When you treat with an antibiotic, you change the shape of the microbiome, and in some instances, if that does not recover, it may predispose an individual to other more severe forms of illness, such as diabetes or, in some instances, obesity. And again, looking at the bacterial populations in these different domains has become now possible by way of the technology advances from whole genome sequencing. So to conclude then, what can we do about this? Well, I think there are a couple of key take-home messages that I'd like to leave you with. The first is, in terms of using antimicrobials to treat anything, we should first of all consider other options. Do we actually need to use a drug? So this brings to mind this idea of prudent use. We should be able to implement measures to reduce the dissemination of multi-drug resistant pathogens through the food chain by way of advocating improved hygiene, better biosecurity, and certainly advocating the use of vaccination. Rosemary will talk when I leave the podium about surveillance systems, but I think surveillance systems are critically important here, particularly surveillance systems that are designed to link what's happening in animals with what's happening in humans, and I could add here what's happening in the environment. And there are examples of some of these. The DanMap one I particularly like, and John mentioned that, and I wish we had it here on the island of Ireland. We could also improve education, training programs, and engage with the public. And there will be people here talking about consumer issues reflecting this issue also. John and others have mentioned research needs that we need to prioritise. We certainly need to develop new drugs because, as I've said, there are no new drugs, so what we have, we hold. We need to look at treatments and diagnostics. And I think meetings like this network meeting will help us to strengthen collaboration on the island, but hopefully internationally as well. So to stop, I would say, antibiotic resistance is no longer the preserve or confined to hospitals. It's here in other environments as well. The bacteria associated with animals, food and water are major sources by which resistance can be transferred to humans. And we need to start to develop a really detailed understanding of how these mechanisms occur and develop controls to stop them. Thank you very much.